Hello, this is Dr. Jabour, and this lecture will be about lipids and the cell membrane. So let me close this. We actually don't need that. There we go. So before we get into the lipids and how they make up the cell membrane, let's make sure that we know what is a cell. So we have a number of things that are actually non-living. They're considered, non-living. They're like atoms and molecules. So in the lecture on basic chemistry for biology, you learned about, let me grab this. You learned about elements in the periodic table, element of carbon, element of oxygen, and within each element you have a number of atoms. They're all carbon, but they vary by their atomic mass. And see these atoms, they come together to form molecules, and that's what you see over here. So an atom has a nucleus that's made of protons and neutrons and has electrons that are gravitating around the nucleus. And then atoms come together and they form bonds between each other, just like the bonds here between these two carbons, these two carbons, these two carbons, etc. So we have molecules that are formed from the addition of atoms together, and then these molecules can be really big molecules, and they form these four macromolecules that we've seen in previous lectures, like the nucleic acids. This is what this is. This is actually a nucleotide, the monomer of nucleic acids. Here it looks like it's a chain of amino acids. This looks like a chain of glucose that forms glycogen. And this is actually a DNA molecule that is made of many, many nucleotides. This looks like a phospholipid. So the molecules are non-living, and they come together to form a membrane, to form different little, we call them organelles. They're like little organs inside of a cell. And we'll talk about organelles, in a separate lecture in more details. But they form all of these molecules. They come together to form like little boxes that will be inside the cell. And the membrane will be the limit, the edge of the cell. So atoms, molecules, and organelles are non-living. On their own, they can't, nothing's going to happen. However, when at the level of organelles and when we have a membrane around all this and we have a cell, that's your unit of life. That's the smallest unit of life, the cell. See, so atoms, molecules for molecules, then macromolecules are really large molecules. They all come together to form these mini compartments inside the cell. And then the cell is your first living entity. And then the cells, they can, match next, bind next to each other and form tissues. We call those tissues groups of cells. And tissues together will form organs, and organs are part of organ systems. And this is basically the transition all the way to an organism. So let's get back to the plasma membrane. The plasma membrane is the edge. It's the boundary of the cell that separates the cell from its surrounding. It is very important that you understand that it's a selectively permeable structure. It's permeable, meaning it does allow things to go in and out. So it's not like a brick wall. It is permeable, but it's selectively permeable. So only certain molecules will go in, other molecules will not be able to go in. Certain molecules will just kind of flow in and out, but other molecules actually need a specific transporter on the membrane. So it's selective. So it allows some substances to cross more easily than others. We also call it a fluid mosaic model because it's fluid, meaning once all of the components that make the membrane are together, they're not stuck there. They're actually moving within the membrane. For example, proteins in the membrane, they can flip-flop in the membrane. So it's actually fluid. We consider it to be fluid. And mosaic, because it's actually not only made of lipids, but there are proteins embedded in it. We talk about that in the protein lecture. And there are carbohydrates on the proteins and carbohydrates on the lipids on the membrane. So it's mosaic, and there's all kinds of different proteins embedded in there. That's why we say mosaic. That means all kinds of different things. And that's the similar diagram that I used in my protein presentation, where at the time I talked about the integral proteins in the membrane and the peripheral proteins that are underneath. And all of this is quite, fluid. And you see it's kind of like, it's bent like this in the drawing to show you it's not stiff. That's the idea here. And there's many things, right? So there are these phospholipids, and this is what we're going to talk about today. These are lipids, and basically they form the core of the membrane. That's basically the most abundant molecule in the membrane, the phospholipid, because it makes the membrane and then other molecules are embedded in it. So the phospholipid bilayer is possible because phospholipids are amphipathic molecules. Amphipathic. What does that mean? It means they have two sides. They have a polar head and they have a non-polar tails, or we also call them hydrophobic tails. These are hydrophilic heads. So hydrophilic, hydrophobic, this was discussed in the lecture of chemistry for biology. We talked about the polarity of molecules. When the electron cloud is kind of pulled by one of the atoms more towards that side of the molecule becomes polar. It has one side that's a little bit more negative than the other. And that allows the molecule to form hydrogen bonds with the water. So these polar heads, there's a polarity to them, so they can form hydrogen bonds with the water molecules that are in these environments inside and outside of the cell. These are aqueous environment, that means it's water-based, and water is a polar molecule. Water is slightly negative towards the oxygen side and slightly positive towards the hydrogen sides. And that delta means slightly, right? It's not like an ion where they would be separated and with a full charge. This is just the cloud that's being kind of sucked up more towards the oxygen. So we end up with kind of this slight polarity. And that slight polarity is enough to make these bonds with another polar molecule. So you need to be polar to be facing water. So these polar heads are perfect setup because they're facing aqueous environment on both sides. And the tails are facing each other, hydrophobic with hydrophobic, and they kind of like, kind of like hiding in there and making up the core of this membrane. So in the lipid bilayer, the hydrophilic heads of the phospholipids interact with water, while the hydrophobic tails interact with each other and minimize their contact with water. Okay. So what are phospholipids? They're lipids. So it's and lipids are in general hydrophobic molecules that are rich in hydrocarbons, which means carbon, 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 and hydrogen, 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 hydrogen. This is very non-polar because none of these atoms are pulling on the electron clouds in the covalent bonds, because there are no leftover electrons that are just, not involved in anything. So they're basically, they basically kind of like create this pull of the electrons towards them. But here, that's not the case. There is there's none of that. And if this is still not kind of clear for you, either review the lecture on chemistry, the basic chemistry for biology, or you can reach out to me and we can review that together. So the structure of phospholipids is specific to phospholipids. There are two fatty acids that are attached to one glycerol backbone. And then on the third carbon of that glycerol backbone, you have a phosphate group. So let's see, here it is. Here's the drawing. Well, it's a very nice diagram. So here is the, right here. This is, oops. Yes, actually I was kind of correct. This is the glycerol backbone, right? I usually draw it kind of like, I draw it vertical when I do it on piece of paper. I draw them vertical. It's like, there's your backbone, and then we're going to attach fatty acids to two of these carbons. So fatty acids, I'm going to draw them in orange like it is here. Ding, 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 ding. So these are the fatty acids. And what these are, these lines like this, it's representing carbon, 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 carbon. Every point is a carbon. And when there's nothing else, we assume that this is hydrogen with it. So CH2, 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 CH2. We just don't write it. We just kind of draw these lines with these points. And when you see a kink, that's because there are less hydrogen on these carbons, and there's a double bond between two carbons instead of maximum hydrogens there. So some fatty acids are without double bonds, and some fatty acids are with double bonds. And usually the ones with double bonds are more fluid, and that's the kind of fatty acid in oils and different vegetable oil and olive oil and these oils, as opposed to the ones that don't have double bonds are more stiff, and those are the fatty acids you find in butter or fat in meat, right? After if you pan fry a piece of steak and then you have dinner and then you don't clean the pan, and the fat from the meat kind of just stays in the pan and it solidifies. So there's kind of like a layer of fat that's solidified at the bottom of your pan. It doesn't stay liquid. So these are the ones, the fatty acids with no kinks in them. They're kind of more rigid, types of lipids. On the third carbon, so you see there's this carbon, and this carbon gets the fatty acid. On the third carbon, instead of fatty acids, we have a phosphate group. So usually we just throw a P and a circle around it. But if we could look inside, it's actually a phosphate with oxygens attached to it. And there's even a nitrogen over here. And as you know from reading the previous lecture on chemistry, these are very electronegative atoms. They pull on the electrons that are in those bonds. They're pulling them towards them, and it creates a polarity to this end of the molecule. So that's why this is the polar end of the molecule, and these are nonpolar. All right. So the glycerol is a 3-carbon alcohol because these are hydroxyl groups. And this is also something that we've seen in a previous lecture. So we have a hydroxyl group, another hydroxyl group, another hydroxyl group attached on these 3 carbons. This is the structure of a glycerol. And the glycerol is going to, the glycerol, now the glycerol is vertical, kind of like how I draw it normally. So these are your 3 carbons, and these are the 3 OH groups, right? The hydroxyl groups. So what happens is that 2 of them are going to get basically broken off. And I really shouldn't have done this zigzag here because this looks like a fatty acid. I'm going to do it in green and and do this instead. So what we're going to do is we're going to break these bonds. Oops, I lost my image. We're going to break these bonds and add the fatty acid. So there's a chemical reaction. What is the fatty acid? It's a long chain of carbons and hydrogen, which is kind of what I explained a few slides ago. It's a long chain, carbon, hydrogen, except at the end of it, there is a carboxyl group. So we're going to use that to our advantage to help attach the fatty acid onto the glycerol backbone. And this is what happens. These are the fatty acids. This is the carboxyl group. And this is the hydroxyl group. So now if we want this to bind with this and create a covalent bond, we need to take some atoms out because there's no more room to add anything. So we need to take some atoms out to kind of click the fatty acid onto the glycerol. And then on the third hydroxyl group, we have the phosphate group that's going to be attached on there. So this is what happens. And by the way, this is a dehydration synthesis. And you can see here, we took out the OH. I'm going to do it in green here so you can see it comes from here. It used to be COOH. We took the OH out, and then we took the hydrogen out of the hydroxyl group. So this was an OH over here. We took a hydrogen out, and this was an OH with the carboxyl group, and we took the entire OH out. So H and OH make up H2O. So we've seen this in the protein lecture where I was describing how amino acids come together to form long chains that are called peptides or proteins. And it's done by dehydration synthesis, where we take OH and H off, and basically we create a water molecule that we took out. That's why we call it dehydration synthesis, to be able to connect these two molecules together, the two amino acids in that lecture. That's what I was explaining. Here, we're doing the same thing. It's the same process to create a bond between these two molecules. So fatty acids can be saturated or unsaturated. And I kind of jumped the gun on that because I kind of alluded to that in one of the previous images. The word saturated, and so in your diet, you'll hear maybe your doctor say, try to reduce your intake of saturated fats, and you want instead to use unsaturated fats in your diet, like oils instead of butter. Because health-wise, saturated fats kind of create plaques in your vessels, and they're just not as healthy for you. So what does saturate, the word saturated means? It means it's saturated in hydrogen. There's the maximum hydrogen possible between all of these carbons and on all of these carbons, as opposed to unsaturated. Unsaturated refers to the fact that two of these carbons, somewhere in the middle or wherever, two of these carbons, they actually ditched two of their hydrogens and instead created a bond between them. So, so you end up with less hydrogen than normally you would in a saturated fat, and you have a kink. That double bond creates a kink. You see how the kink here? So that double bond creates a kink in the molecule, and it bends. And the more double bonds there are, the more, of course, unsaturated it is, and the more fluid the lipid is, and the better. So here is a comparison of the two molecules next to each other. So another lipid that you'll also find in the membrane is called cholesterol. So cholesterol is built differently. Cholesterol is considered to be a steroid, and steroids are characterized by the way their carbons are linked together. So instead of one long chain of carbon, 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 we actually have 4 rings of carbon. See, every corner is a carbon. So this would be a CH2, CH2, but we don't write the H's anymore. This would be a CH only because there's already 3 bonds here. So there's only one H possible over here. So you have all of these are your carbons and hydrogen, but it's in ring form. So we have 4 of these rings, and then we have functional groups attached. This is a methyl group, another methyl group, methyl groups, and this is a hydroxyl group. So that hydroxyl group makes the molecule polar just because of that hydroxyl group. Remember, it's because of the oxygen being there. The oxygen is a electronegative atom, and it will pull on the electron cloud towards it and create this slightly negative end to the molecule. And yeah. So here we go. There's the cholesterol in the membrane. The cholesterol is embedded in the membrane right here, right here. So I use red. You see that? We have a cholesterol over here. We have a cholesterol over here. So if we zoom in, you can see, oops, I lost my picture again. You can see, I'm going to zoom in so you can see. You see, these are the 4 rings, the 4 rings right here, and 4 rings. And look. This is very interesting. Look, I'll use my thinner. You see this over here? You see that little gray ball over here? That's your polar hydroxyl group. And it's perfect because this is the part that faces the aqueous environment. So cholesterol is not buried in the membrane. It's between phospholipids, and then it has its polar little head poking to the outside or to the inside. You see, this one is towards the inside. This one is towards the inside. And you can see that polar head facing the inside of the cell. So cholesterol is on facing either side of the membrane, but never buried in the membrane. It does have this polar end to it that gives it this property of being able to face the aqueous environment. There we go. And everything else is very hydrophobic. CH3, 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 CH3. All these are just carbon and hydrogen. This is very hydrophobic, so perfect to be embedded in the membrane. All right. So that's what we looked at. So the fluidity is just referring to the fact that nothing is locked in place. It's very fluid. There's no rigidity. So that depends on the temperature. The temperature will have an impact on membrane fluidity and the degree of saturation of the fatty acids. So the more unsaturated they are, the more fluid it can be, and how much cholesterol is in the bilayer. So this is a great visual here where you can see, if you see kinks in those fatty acids, it's basically going to create some of these spaces and create this more fluid membrane overall, as opposed to more tightly packed, very kind of stiff fatty acids, no kinks, and it just creates a more viscous membrane. So saturated animal fats are solid at room temperature, like lard and butter. Unsaturated fats in plants and fishes usually are liquid at room temperature. You know, like olive oil and fats, and some fats in fish. And then you have trans fats. Trans fats are unsaturated fats that mimic saturated fats. So those are actually not that great, because I think, if I'm not mistaken, those are engineered. I'm not too versed into trans fats. They're engineered basically to taste like saturated fats, but to still have that kink. So, but they're not good for you. Okay, that's what I wanted to say about lipids in the membrane. You have to know how phospholipids are structured and about cholesterol and the properties of the plasma membrane. Thank you.